everyone. I'm DS Stuart Ramage. I've been called to this murder scene as the senior investigating officer. So, on one morning, a man called the police informing us that they'd located a dead body. When he was out walking his dog, they came across this deceased woman, and upon locating the body, it was highly suspected that there was foul play. So we asked the scene of crime officers to attend the scene, and here you have one of our scene of crime officers, Sam Walker. So as there's a dead body in the scene, I asked a forensic pathologist to attend the scene to examine the body, and they indeed have found signs of a struggle and that this individual was potentially murdered. So the forensic pathologist has removed the body and taken it back to the mortuary for a post-mortem. Speaking of which, pathology report already. So from a super quick analysis of the body, is found signs of localized bruising in the front and sides of the neck. He's also found finger point bruising on soft tissues around the neck, a broken voice box, a tiny abrasion over the lower jaw and neck, a widespread superficial bruising and abrasion, and his professional opinion is that there was manual strangulation. Therefore, the police will now look at this as a homicide. As there was blood and other biological fluids on the scene, we're going to ask a forensic biologist to attend the scene to examine and see what evidence can be recovered. So I'll pass you on to Wendy Deegan, our local forensic pathologist, or biologist even.
Thank you, DS Ramage. That's very kind of you to wake me up in the middle of the night and drag me out to a scene. So as you can see, there is a cause for a forensic biologist to come along to the scene of the crime. It was obvious from the start that there was the potential for foul play. And what you can see here is that there is some blood staining around the face of the deceased individual here. So the first thing we're going to do are take some samples to address what might have happened, who might have been there, who might have touched the deceased. But before we get into it all, I feel like I have to address one or two things. I'm pretty sure that most of you have had some little bit of experience with forensic science from TV shows or films. Is that right? Yeah. So you've seen people attending the scene of the crime looking like this, or like this, or even like this, a little bit better. At least we've got a crime suit and maybe one pair of gloves between them. But as my colleague Sam Walker ably demonstrated, this is not what you look like when you go to a scene of a crime. In actual fact, we look like Sam. So we have to be fully protected. And I'd like to say that we're protecting Sam from the samples, but actually we don't really care about Sam. What we're protecting are the samples and the scene of the crime from Sam. We don't want the individuals to be contaminating the scene of the crime. We need to make sure that if you're going to cough or sneeze, you do it inside your mask. We don't want that happening all over the scene of the crime. So we're going to have our scene of crime suits. We'll have masks. We'll have the hoods up. We will be, as Sam will tell you, double gloved to make sure that nothing is getting out there. We will have shoe covers on to cover our shoes so that nothing that is on our clothing will be taken into the scene of the crime and potentially contaminated. It's not glamorous. We don't go running around the countryside toting guns and a full face of makeup and high heels, unlike in the TV shows. Nobody can see who we are behind the mask and the hood and the safety glasses. But this is the reality. Sometimes finding the evidence and finding the DNA can be a challenge. Now, that might be because things look quite clean and there isn't anything obviously apparent. There's no staining, there's no obvious evidence to collect. Sometimes finding the DNA can be a challenge because it looks like this. Now, the reality is a scene of crime is much more likely to look like this than the previous slide. In fact, this one's mild. There's some I wouldn't want to show you. So what you can see here is a lot of potential evidence. There are cigarette ends, there are more cigarette ends, there are drinks bottles galore, and there is a myriad of body fluids over in this region here. Now, obviously, sometimes at the scene of the crime, we have to weigh up what we're going to collect and what we're going to leave behind. We don't want to miss an opportunity to pick up evidence. If you don't pick it up when you first go to the scene of the crime, you may miss your opportunity to collect it at all. So we do have to be careful. And you can see at the scene of the crime here, we have started to place markers at points at the scene where we think something might be evidentially significant. And once we've done that, we're going to start actually collecting the evidence. It would all, as you saw Sam doing earlier, we would photograph it with a scale and with a marker, and then start to collect the evidence. So at the top here, you can see at the scene of the crime, near where the body was, there's a cigarette end. Now, this might be relevant to the crime. We don't know at this stage, but we're going to collect it and we're going to take it back to the laboratory to do DNA analysis on to see if we can find out who might have had it in their mouth. We have a can of Diet Coke here. We'd also want to take this back to the laboratory and we could swab around the mouth of the can to try and detect saliva from anybody who drank out of the can. Sometimes at the scene of the crime, we find slightly random objects that we don't know what they are, like we have here. But we're also going to collect these and take them back to the scene of the crime just to see what it is. Also at the scene of this crime was a condom. Now again, we don't know if it has been related to the crime or not, but certainly we want to collect that as potential evidence. We don't know at this stage whether there is a sexual element to the crime, and we don't want to miss our opportunity to recover the evidence. Thank you, Sam. So here we have our victim lying in this outdoor scene.
Thanks very much, Wendy. We've now collected a very important evidence, the scene. But as you've noticed, this is an outdoor scene, and what may not commonly be known is that there is a plethora of potential evidence which can be collected from the outdoors. So what we're going to now do is call on a forensic soil scientist to come to the scene and assess what potential evidence could now be recovered. Lorna, Dawson.
Thank you, DS. Thank you, boss. So, soil, you might think, that's just dirt. What is it? Why is it connected with forensic science? Well, soil is one of the many types of trace evidence that can be used to try and test a potential link between a perpetrator and a particular scene, or indeed test it as compared to an alibi location. And it's like blood, hair, fibre, plant fragments, pollen, glass, all those things, as Wendy said, can be taken into a scene or out of a scene. So that principle of every contact leaves a trace was Edwin Lockhart in 1910, and that still persists to today, that we use that principle and try and test that relationship between an unknown sample, that's one where we don't know its origin, and compare it with a known sample that we know where it's been collected from. Soils are everywhere. Soils are different. They vary in many, many ways. We all have to get around by car, by feet, by bicycle. We have to move around the countryside and we pick up soil. Have a look at your footwell mats in your car. Just think about that, what's there. You know, there's always material. Have a look at your shoes when you go home. You'll be able to see that you bring back soil from places you've been to. Particularly when it's wet or peaty or clay, the soil sticks. And even if you don't know, you've got splashes on your trousers. And we've now got methodology that we can work with tiny traces of soil that you, as a perpetrator that's been somewhere, will not even know that you've taken out of that scene, and can be transferred to clothing, footwear, vehicles and tools. All of that can be looked at to try and test for a potential link with a potential location. So back to the murder. And here we've got a victim with marks and actually debris under their nails. That is a key area that we would want to sample, because we want to compare the characteristics of that debris under the nails with the potential soil and vegetation at that particular scene to see if the struggle has happened there or potentially somewhere else. But of course, before that's done, the biologist takes the swabs for the DNA. And once the DNA has been done, the other types of evidence is collected. And we'd also look at particularly the footwear and the clothing. And we'd look at the soles of the footwear, because that tells us about where the victim has recently walked and where they've come into contact with soil and vegetation. But before we decide on how we're going to sample our known samples, our samples to represent the crime scene, we have to make sure, as we were shown by DS Ramage, that there was a common approach path. So the scene has been secured, double cordoned by the police, and a common approach path. That's the path that is taken by all the people, the SOCOs. Everyone that visits the scene must be kitted out in full PPE. And they mustn't walk on an area that might be crucial for taking the reference samples. So that's why the common approach path is defined, and any personnel is logged in and out and must walk along that path. And the questions that the DS was asking was, would they have had access? Would the offender have had access to a vehicle, or was the victim able to walk and run? Is there evidence that the offender would have contacted the ground other than by standing or walking or running? There's other ways that people can arrive in a particular location. All these questions are going through the investigator officer's head. And we, as forensic scientists, are there to help provide the evidence and clues for them to work out what happened. We're servants of the Crown when we're invited onto the crime scene. It's a very privileged position to be in. But as a forensic soil scientist, we have to look, along with the intelligence that we're given by the investigating officer, if there's no CCTV image or there's no witness evidence that the car was parked here, the suspect vehicle, then we have to sample in all potential entry routes and exit routes, because we don't know yet how they got there and what vehicle was used. We have to sample at potential contact points. Here we've got a lay-by near to the scene. There's marks there, vehicle marks. So do we sample them? Are they cast by the SOCO? And do we sample at different depths, depending on the moisture condition at the time? We would take contact point samples, so those where there's any potential marks that there's been a recent disturbance by foot or by vehicle. But if we don't have that information, then we would sample in a strategic way at any pathway or any route around the body. And the sampling has to be done in a proper protocol that is a standard operating protocol so that it's reproducible and repeatable and done by the SOCOs but under the experts' guidance. So here, for example, we see Sam, the SOCO, taking the careful with double glove full PPE with a scale, a marker, and then collecting that sample into an appropriate vessel. In this case, it's a pot with a lid, so that the material in soil, for example, we get stones and grit. And if that went straight into the plastic evidence bag, it could potentially breach that evidence bag. And that's one of the crucial things with any evidence in a particular case, is that they have to be carefully packaged in an appropriate package. And in this case, it's a sealed evidence bag. It must be formally signed, dated, corroborated in Scotland, and also the packaging should be checked when it's passed from the field and submitted to the laboratory, that all of that information is correct at the time. Because there's no point in doing all the fancy analyses if you find that then it's submitted as a production to court, that it's thrown out of court because there's a signature missing, there's a date missing. So that's as important as any of the other procedures that go on at the scene. So that at this point, I now hand over to the boss here, who's going to tell us the next stage of the inquiry.
So our experts have attended the scene and have collected all the evidence that they deem to be able to help with our investigations. So the next stage is we're going to ask the forensic scientists to get us back to their laboratories and analyse this. But first, what we do is we sit down with the experts and have a forensic strategy meeting, basically just to talk about what evidence we have, what potential it could have to us in our investigation, and how they'll proceed in analysing that evidence. So we're going to first submit the DNA and the fibres to Wendy Deegan's lab to look at the evidence that's collected.
Thanks, Sergeant. Thank you very much. So, as the DS has said to us, we've collected all the evidence, and I'm going to take it back to my laboratory to see what we've found and what the significance of that evidence might be. And the first thing we're going to do is look at the fibres evidence. So if we suspect there's been contact between two individuals, it may be that there has been a transfer of fibres from one individual's clothing onto the clothing of the other individual. So in this photograph here, you can see one of my colleagues, and they're examining the pair of jeans that you saw the victim wearing, looking for any potential fibres that might have transferred from the perpetrator's clothing onto her jeans. And this will be done in a very methodical way, taping areas of the jeans piece by piece, and then looking to see if there are any apparent foreign fibres that don't belong to the garment itself, or indeed to the other garments that the victim was wearing. So ideally, what we're looking for is when we have an item of clothing from a perpetrator and an item of clothing from a victim, we're looking for this transfer or potential transfer of fibres. And finding fibres that have transferred from one garment to another is excellent evidence to show that there's been contact. And seeing transfer the other way from the victim's garment back to the accused garment gives us even more significance of that evidence if there has been two-way transfer. In this case, what we found on the victim's jeans were some dark blue woolen type fibres, which were obviously foreign to the garment itself, and in fact didn't match any of the other items of clothing that the victim was wearing. So they do appear to be what we would class as foreign fibres, and therefore they're of particular interest as maybe being from a garment that the perpetrator was wearing. Now, to get on to my favourite subject, body fluids. As a forensic biologist, I love a good body fluid. I love them all. And when we get items submitted to the laboratory, that's one of the first things we want to see, is which body fluid is it? And we have tests we can do. They're called presumptive tests. They're quick, easy tests that don't require any fancy equipment or instrumentation. They generally rely on a colour change. So we can do them at the scene of the crime or in back at the laboratory. They're quick, they're easy, they don't take a lot of equipment. So we can do these for saliva, for semen, and for blood. So let's have a little look at this. The first one we're going to look at is the saliva test. So we collected, or Sam collected, this can at the scene of crime for us, this Diet Coke can. And what we wanted to do was look at whether we could detect, first of all, detect saliva on the rim of the can where somebody might have been drinking from it, and then look to see whether we could do DNA analysis and obtain a DNA profile from that individual. Now, the test that's most commonly done to detect saliva is called the Fadabas test, and using a reagent that in the presence of saliva, it's actually the amylase that's in the saliva that causes the reaction. It will give this lovely blue color, which is distinct from the background, and will let us know that that is a positive result for the presence of saliva. And that's what we found here for the Coke can. It gave us a positive result. So we know that somebody drank out of it, and therefore we could swab it and send it for DNA analysis. We also have a couple of tests that we can do for the presence of semen, the most common one being the AP test, the acid phosphatase test. It is also a color change test. So if there is semen present on an item, it will give us this purple color. Turns to a bright purple color, indicating the presence of semen. And in this case, we didn't find any semen present, apart from on the condom. That was the only semen. We didn't find any semen on the body itself, on the deceased, or from the intimate swabs. The only semen we found was on the condom, which had been recovered from the scene. When we recover an item that gives us a positive AP test that suggests that there is semen on an item, we always want to check that it really is a genuine result. And the confirmatory test for semen would be to make up a microscope slide, stain it, and we would be able to look for the presence of spermatozoa. Now, what you can see on this slide are these large pink cells here with the purple nucleus. So these are epithelial cells, these ones here. And these little dark purple critters here, these are the spermatozoa. So not always very easy to find, but that categorically shows that it was a semen sample. Now on to the forensic biologist's bread and butter. If there isn't blood in the case, we're always a little bit disappointed. We're a weird bunch, it's true. So here we have a couple of the tests that we might do for the presence of blood. These are presumptive tests that again rely on a color change. We have the Castle-Mayer or KM test, which turns a nice pink color to the presence of blood. This is the one, if you've watched a TV show where they're doing silent witness, they always use it. And they take their swab out of their scene of crime kit, they give a little rub onto the red staining, and they get a little dropper bottle, drop one drop of liquid on and it turns bright pink. Who's seen that one? Yeah, I've seen it too. Don't get me wrong, I love silent witness. The test is wrong. It's not how you do it. They're close, but they're not quite there. You actually need to drop 2 chemicals on before it should turn pink. But I guess for speed on TV, there, just do it quicker. So there's our KM test, which will give this lovely bright purpley pink color if blood is present. Or we can use the slightly tougher one to say, the leukomalachite green test, the LMG test, which, as the name suggests, turns green with the presence of blood. Nice, bright, bluey green color showing you that there's blood present. Now, when we can see red staining, these are the tests we can do. So a nice little swab of the area of red staining, drop your chemicals on, it'll turn pink or it'll turn green. What if we're at the scene of a crime in a house, say, and we're given the information that a body was found nearby in a wooded area, but the thought is that perhaps the crime occurred here at a suspect's house. So we go to the scene of the crime, we go to the house, we start looking around, and we can't see any blood. There's no apparent blood, There's no obvious red blood staining. What do we think? Well, there's two things. Either the information is all wrong and the crime didn't happen there, or maybe what's happened is there's been a cleanup and that the blood that has been on the carpet or the floor or the wall has been cleaned up. And you can do quite a good cleanup attempt so that visually you can't see any blood remaining and the perpetrator thinks, excellent, I've got away with it. We do have a technique that we can use to try to see the blood. And again, I'm sure you've seen this one on the TV shows and the films, luminol. And they go in and they spray a little chemical around the room and suddenly it glows a white blue color. They do it wrong as well. But it's the same result. If we, at this particular area here, there's no, despite the slightly 70s, 80s swirl of the carpet, there was visually no blood there. Once we darkened the room and sprayed luminol, it looked like this. So what you can see here is not that there's actually significant blood staining there. So we know there was loads of blood seeped deep into the fibers of that carpet where the cleanup attempt didn't get to. But it doesn't just tell us that there's blood there. It also tells us something about the crime. And this is why I like this photograph. It's so informative. It shows us, in fact, where the bleeding head of the deceased went bump, 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 bumpity bump all the way down those stairs as they were dragged out of the house. So it's really informative. We can also tell something by the pattern of the blood. So if we have a scene of crime where there's blood on a flat surface, on a wall, on a floor, maybe even on a ceiling, we can tell a little bit about how the crime happened, what might have occurred. So we might see something that looks like that, this. This is impact spatter. And this is where an object that is wet with blood is hit with force, and that blood will spray outwards onto a surface. So that's impact spatter. We might see expirated blood, which, as the name suggests, is where blood is in the victim's nasal passages or mouth, and as they breathe out, tiny little particles of blood will be breathed out. We might see arterial gush. This one is obviously where an artery has been severed and the blood is still pumping out, and you can see where it goes up and down as the heart is pumping until eventually the heart sort of runs out and it stops. Or lastly, we might see something like this. Now, this is cast-off blood. This is where we have our item that's wet with blood, let's say a head, and I have a weapon, let's call it a hammer, and I'm hitting that head or that item that's wet with blood. Now, you imagine, once my hammer goes into that once, the end of my hammer is wet with blood. As I take it back to go for a second blow, I've just cast off blood from my weapon up into the ceiling and probably to the wall behind me. And it gives quite a distinctive pattern that might look a little bit the same as this one, but it isn't. It's quite distinctive. And a blood pattern analyst will be able to tell exactly where this blood has come from. So highly informative. Let's go back to our scene of the crime. So there wasn't any elaborate blood pattern analysis in our scene, but we did have quite close to the victim where she was lying. We did have a stone with a bit of a jagged corner here and a significant amount of blood on that stone and running down the stone towards the ground. Once we've collected all these body fluids and we've tested them, we then obviously want to do some DNA analysis. So we want to try to work out whose DNA is it? Whose blood is it at the scene of the crime? Whose semen was it? Whose saliva was it? So we do DNA analysis. Now, this is what a DNA profile looks like. Once it's been through the whole process of extracting the DNA and amplifying the DNA up to look at the very particular parts of the DNA we're interested in, what you can see here is a DNA profile from the technique called global filer. And that's the technique that the Scottish Police forensic laboratories use. It looks at 24 areas of DNA. So as I'm sure you all know, everybody's DNA is different. We all have differences in our DNA. We're all unique, apart from identical twins. So if anyone is an identical twin, then you and your identical twin will share your DNA profile. But we don't look at the entirety of your DNA genome. We just look at these specific 24 regions of the DNA. And of these 24 areas, 21 of them are areas called short tandem repeats. And these are areas that we know vary greatly between individuals. And they're areas that allow us to tell you apart. And then the other 3 areas are areas that will identify the genetic sex of the donor of the stain. So that's a global filer profile. Now, what we're going to show you tonight is that the DNA can really be broken down into something very, very simple. That DNA consists of just 4 base pairs, an A, a C, a G, and a T. And the entirety of our DNA genome and all the differences between us, all the visual differences between us, are due to the differences in the patterns of those 4 bases, the A, the C, the G, and the T. And this is representative of the DNA we found that must be from our suspect and was found on the victim. Once we've done our DNA profiling and we've identified a DNA match, we then need to give the court an understanding of the significance of that evidence. And so we calculate a statistic. Now, it's quite complicated how we do that calculation, but at the end of it, we would come out with a statement, something like this. The DNA profile is 1 billion times more likely to be observed if the DNA came from the suspect rather than from an unknown, unrelated person. And just to point out that we are therefore looking at the DNA from the point of view of the prosecution, suggesting it was the suspect, and from the defense's point of view, which is from an unknown, unrelated person. So we're keeping a very unbiased viewpoint. We don't work for the police. We don't work for the prosecution. We're there to deliver the evidence that we found from the analysis that we've carried out. And I'm going to pass you back to DS Ramage to take you through where we're at in the investigation.
Wendy's taken back the biological evidence to help us address the who questions, as in like who is involved or who potentially committed this crime. We also have our soil and vegetation evidence. So while that's ongoing with Wendy, we're going to submit these samples to Professor Dawson's lab to analyse and get back to us what information she could use to help us answer the where questions. Was this crime committed where the body was found, or was there potentially a second crime scene?
Thanks, Deegan. So, as yes, Ramage said, the forensic soil scientists or forensic botanists, they're helping answer the where question. Where could that soil or vegetation have come from? Now, we answer that question from 2 main aspects. One, to help them narrow down and exclude areas that it has not come from, to prioritise areas to really search and put the valued resources into working out where the crime was committed. And on the other side, it's the trace evidence comparison. But what we've got is a range of different evidence types. So questioned items, as I said earlier, could be vehicles, they could be clothing, but one of the main types of item we get submitted to our labs are footwear, because you can't avoid contacting other surfaces and transferring material to your footwear. We sample that in layers. We sample it in particular locations. So these are all images from actual cases. And you can see here, there's a lot of soil on the front of this shoe. Now, this was a suspect shoe in a rape case. And when we got this submitted to us, we said it's obviously quite firmly in one position on the item of footwear. And then the SOCO team went back to the alleged scene, and they went and saw at the location there was little marks similar to the indentation in this particular item of the footwear. And in that case, we got two semi-independent pieces of evidence that linked the person wearing that shoe to the particular scene of the crime. We got the soil that had the same characteristics as the soil at the scene, and there was also the casts made of the impression in the border that matched that particular mark on that shoe. So two different pieces of information that linked the person whose DNA was inside that shoe and wearing it at that time. And then here you can see this, there's masses about of vegetation. There's deciduous leaves, there's grasses, there's mosses, there's herbs, and there's soil there. But what we do is we sample those all separately because there could be more than one location represented on that item of footwear. Have we got any gardeners or people that work with soil here? People that love soil because it keeps us alive, it gives us our food, it freshens our water, it cleans our air. We couldn't live here if it wasn't for soil. This is a thin section through soil and magnified up hundreds of times, but this is pore space. So we need that for the water to move through the soil to keep it fresh for growing crops. These are what's called clay coatings on that pore. And that gives nutrients to the growing crops. There's minerals there. And these minerals are from the bedrock or any fertilizers that have been applied. Now, the great thing is that we've got all that geological information mapped. We have that spatially referenced. And every mineral suite that you get within the soil relates to the minerals that you get in that geology. And that's all georeferenced. So that tells us about the inorganic part of the soil. And then we've got the organic part of the soil. That's the organic matter that builds up. And that builds up from all the material that breaks down and grows there. It breaks down and leaves behind the recalcitrant, the non-decomposing part. It leaves behind chemical biomarkers. We can analyze those biomarkers, and from that analysis, we can tell you what's grown at that unknown location. With that information, plus we now are able to look at bacteria, fungi, mites, you name it. We can sequence the DNA of all the organisms that are in the soil. A nice little fact that a handful of soil, there are more organisms in that handful of soil than there are people living on the planet. I think that's absolutely incredible. So we've got there loads of information that we can potentially use. Anyhow, so back in the lab, the forensic scientist has taken the production out of the evidence bag and sets about the examination and recovery of any material after it's been described, the item itself, or where the material is adhering to it. Then the individual items are recovered into separate production vials. We use a range of different methods, and I'm not going to go into them today, but depending on the size, the condition, and the type of material that we've recovered will dictate which of these many methods that can be applied to the soil and vegetation are used. But back to that geospatial information. When we recover material from the item, we can link it to mapped information. Here at the James Hutton Institute, we produce many maps at various scales, which tell us what kind of soil we have in the whole of the countryside. So we can use that spatial information when we're helping the police work out where a crime was committed. And in this particular case, what the forensic scientists recovered were little bits of grit that had got embedded in the tread of the shoe. Now they were recovered and they were looked at And we knew because there was a spatial boundary to when the victim was last known to be alive, where they'd used a credit card in a town called Inverurie up here. So what we looked at were all the parking spaces, all the areas on the road which had that type of grit in the surface. And these are the blue areas here that we could map. Then we were able to recover areas of peat which had stuck in the tread and analysed that and found its pH was 5.4. So we then could map all the areas that had that same pH. So we're adding layers of information, narrowing down. We could also find the vegetation that was embedded and tiny little specks. And here we've got a Scots pine needle. So we know that the person had recently stood in an area that Scots pine was growing. Also, tiny fragments of moss. There was a recent case about a year ago. There were 6 different species of moss we found on the footwear of the perpetrator, and they didn't know they had anything. It had gone right into the deep cracks of the soles of the footwear. And all that information could then be analysed. So we narrowed it down to this area here at Kirkhill Forest, where you can see there were 3 areas that shared the characteristics of having the grit, having the peat at the pH, and having both Sitka spruce and moss vegetation growing. There might be other things as well, but those were the things that we recovered. And therefore, it took us, or suggested, we could give that information to DS Ramage, who then could go to these areas and assess whether that was a particular location where there might have been an interaction or recent disturbance of the ground vegetation. Yes, did you manage to go to those sites?
Yes, so I asked the search team to go out to these 3 locations, and one of the locations, our search party found signs of a struggle. There's broken vegetation, and we then recovered some evidence from there and submitted it back to yourself.
Was there any signs of biology? Was there any evidence?
There was also signs of some blood as well, which we collected and have passed back to the DNA laboratory as well.
Okay, thanks. Thanks for submitting. We'll just better get back to the lab and get those finished.
Yes, we had.
Yeah. So now it's to the valuation of the soil and vegetation. We're getting to the crux of what does this information that we have been ascertaining from our examination and our analysis. So first I'll do the soil and vegetation, and I'll hand over to Wendy to talk about the fibres and DNA. So what does the soil tell us that we recovered from the victim's footwear? Well, the uppers of that, those boots, had material where she was found. That's not unusual because that's where she was found. There were bits of beech leaves, and they were embedded in the laces. But when we looked at the soles, we found that this peat, matching the peat at the Kirkhill Forest, plus the vegetation at the Kirkhill Forest. So this tells us in the Lockhardt's exchange principle that it's likely that she had stood or walked most recently because it was the topmost layer on the soles of the shoe at this particular location. But apart from the few fragments of the beech material that we found on the laces, there was no other evidence anywhere that she'd walked or stood at the place that she was found. And the fingernails, if you remember at the beginning, a particularly crucial part can be what is embedded underneath the fingernails. So those are clipped and those are sampled under full PPE and sterile conditions. And those clippings, I know they look disgusting, don't they? But we do get some disgusting things, so we can't. Anyway, you forget about that and you focus to be objective. And as Wendy said earlier, no matter who is commissioning us to do the work, we've got to be independent, objective, and impartial. So whether it's the prosecution or the defence, we have to present what we find. Irrespective of what we find, those are the facts, and our duty is to the court. So in this particular case, here we see that there was this peaty material which had the same chemical characteristics as that of the Kirkhill Forest. So to summarise, rather than a quantitative approach, such as the statistics given with the DNA, we say it's much more likely to, or more likely to, or less likely to have come from one location relative to other. It's always given in the alternative proposition approach. So the proposition of the prosecution, which has it's come from the crime scene, but the defence could say, no, it's just as likely to have come from the home address or the place where the suspect works. So we've always got to consider what is that alternative proposition. But the soil and the vegetation on the sole of the victim's footwear, as we saw in the analysis and examination, was the same, that peaty soil with the same pH as that at Kirkhill Forest. Also had 2 items of evidence of vegetation, the moss and the Scots pine needles, as at Kirkhill site. The same was for the soil under her fingernails. And the soil and the vegetation, you didn't know, but as you came in here tonight, you had a artificial intelligence scan of your whole body, including the soles of your feet. And we recovered your DNA as well. It's all right, it's all been anonymised before you go home tonight, so don't worry about it. We also looked at the soil and vegetation on all your feet, your footwear. So we will have that information as well. And to test whether that is the same. But indeed, we're looking for someone that's also been at the Kirkhill site, where the primary murder site, where that was the site where she was attacked. But what we found is on that person of interest here tonight, also found soil that was on the bank at the deposition site. So she didn't stand there, but that's suggesting to us that he likely stood there. Or that's what I would say to you, DS, that's likely what happened.
So to just give you some conclusions and some interpretation on the significance of our findings for the fibres, the blood and the DNA. And again, as Lorna has done, we're using Lockhardt's principle of every contact leaves a trace. So what we have here is we have that swab from the Coke can that we detected there was saliva on. Well, what we found there was a full DNA profile that came from a person of significant interest that was likely to be involved in the crime. We had taken swabs from the victim's fingernails, and we found what we strongly suspect to be the perpetrator's DNA under her fingernails. And we obviously found DNA from the victim was at the scene of the crime on that stone that I showed you earlier that had the blood dripping off it. That was indeed DNA that matched the victim. So just to go through quickly all of the evidence that we obtained, we had an unknown suspect's DNA present on the victim's jeans, and also under the victim's fingernails, and also a swab that had been taken from the neck of the victim. We found, as I mentioned, the victim's blood on the stone at the scene, and at that scene of the crime that DS Ramage brought me out to at Kirkhill Forest, we found the blood of the victim at this disturbed site. Lastly, when we looked at the fibres, we recovered these blue fibres from the jeans of the victim, and we're trying to match that to any of these individuals here tonight that we strongly suspect may be involved in this crime. So I'm going to hand you over to DS Ramage to go through the evidence and see if we can work out who indeed did commit this homicide. Thank you, DS Ramage.
Thank you. And what fantastic effort from our scientists who did all this work from the comfort of their seats over there. We're doing it with some work. So, from Professor Dawson's analyses, she has identified that the person of interest is likely from Aberdeen. So if you'd like to look under your seat, you will find all this evidence very magically and use when you're leaving tonight. So, if your image matches this scene about her being please stand up from please. Rather a lot of you see. So the next piece of evidence that Dawson is informed us of is that there's feet on the floor of the shoes. So if your image matches this one of the feet, you remain standing. If not, please sit down. So if you've got a blank card, which means we haven't been able to recover that evidence from you tonight. But as the scientists have informed me, the absence of evidence is not evidence of absence. So you please remain standing for the time being. Next, there was pieces of moss present on the footwear as well. So if you have moss, please remain standing. If not, sit down. And again, if you have a blank card, remain standing. The next piece of evidence was a Scotch pine. So again, please remain standing if you have that, and if not, please sit down. So on to the fibers and DNA evidence. There was signs of these blue fibers. So if your image matches this one, please remain standing. If not, sit down. You'll be aware there are two blue fibers that this is a thicker fiber we're looking for. So, our final piece of evidence tonight is the DNA. So if yours matches this one with the purple, red, and the orange, blue, please remain standing, and if not, please take a seat. It's purple, red, and then orange, blue.
Oh, it's been red. That's good.
So, you're now down to three suspects. Gentleman in the blue jacket. On the back of your DNA cards, what does it say on it? The lady in the black. So says a friend, there's a likelihood that she would have a reason to have her DNA on the suspect's body, so you have now been excluded and you'll be seated and yourself at the back. Neighbour, again, since there is a legitimate reason for this.
There you go, Sarge.
So, if you are all free to go tonight.